Good day, everyone. This video is part 1 of our discussion in natural products. This will be our outline. First, we will talk about primary versus secondary metabolites. Then 2, I will explain why the focus of natural product chemistry is on secondary metabolites. 3, I will show you sources and examples of secondary metabolites. And lastly, we will discuss the types of secondary metabolites. Metabolites, as the word suggests, are formed through metabolism. Here is a map of metabolic pathways. Metabolic pathways occur in chains of reactions, and different intermediates are formed along the way, as you can see. These intermediates and products are collectively known as metabolites. Many metabolites can range from small molecules with just a few atoms, but they can also be in the form of macromolecules that consist of hundreds or thousands of atoms. Metabolites can be divided into 2 categories: primary metabolites, as produced from primary carbon metabolism, and secondary metabolites, produced from the secondary carbon metabolism. Primary metabolites, as the word primary suggests, means they are the first ones to be synthesized. So they are directly involved in body's basic functions like growth, development, reproduction, and respiration. Primary metabolites have a major role in those reactions, so they are important for growing and keeping the organism healthy and alive. An organism without primary metabolites is like a child that does not receive any food or nutrition. It won't be able to fully mature and will die at an early stage in development. Some examples of primary metabolites are amino acids used in building important proteins, sugars, carbohydrates, nucleic acids that build up DNA and RNA, as well as lipids that are needed in the structures of cells and tissues. Next, secondary metabolites. These are derived from primary metabolites. In contrast to primary metabolites, however, secondary metabolites are organic compounds which are not directly involved in the growth and development of an organism. They are not fundamental for the body to function. Secondary metabolites may be chemicals needed in adapting to the environment in order for the organism to survive. These are most commonly produced by plants, fungi, and marine organisms, seldom lang sa animals. Because animals, when placed in a dangerous environment full of predators, they can easily run away. But for organisms like plants and fungi that can't run away from danger, they resort to chemical means to protect themselves and ensure their offspring. So, secondary metabolites produced by these organisms can be poisonous to deter organisms from eating them. They can also produce substances that make them more attractive to pollinators, and they can also produce chemical signals for communication with each other. Well, meron tayong poisonous and venomous animals that have developed natural products for self-defense as well. By the way, secondary metabolites are also limited in occurrence or distribution because only particular species of organisms evolve to produce a specific secondary metabolite, unlike primary metabolites that are common in almost all living organisms. Secondary metabolites can still include amino acids, but not the ones used in common proteins. These can also include nucleotides, but not the ones in DNA and RNA. And these can also include certain lipids. But these include a whole lot more molecules that you might have never heard before. Let me just show you a couple of videos to differentiate primary and secondary metabolites and to further introduce you to what secondary metabolites can do.
So what is the difference between primary metabolites and secondary metabolites found in plants? Primary metabolites are compounds that help to form the general shape and structure and scaffold of a plant. Examples of primary metabolites are proteins, carbohydrates, sugars, polysaccharides, and vitamins and minerals are also primary metabolites. So these compounds help to support the plant as it first starts to grow. Now, once the plant is growing rapidly and maturing, the plant will then start to produce beautiful organic compounds. These are natural compounds found in plants, also known as phytochemicals or secondary metabolites. These secondary metabolites are produced by plants in order to help the plant survive. Secondary metabolites are also used in plants for defense, for cell signaling, for plant-to-plant communication, and many other processes that we don't fully understand as scientists.
This is a tomato plant, and this is an aphid slowly killing the tomato plant by sucking the juice out of its leaves. The tomato is putting up a fight, using both physical and chemical defenses to repel the attacking insects. But that's not all. The tomato is also releasing compounds that signal nearby tomato plants to release their own insect repellent. Plants are constantly under attack. They face threats ranging from microscopic fungi and bacteria, small herbivores like aphids, caterpillars, and grasshoppers, up to large herbivores like tortoises, koalas, and elephants. All are looking to devour plants to access the plentiful nutrients and water in their leaves, stems, fruits, and seeds. But plants are ready with a whole series of internal and external defenses that make them a much less appealing meal, or even a deadly one. Plants' defenses start at their surface. The bark covering tree trunks is full of lignin, a rigid web of compounds that's tough to chew and highly impermeable to pathogens. Leaves are protected by a waxy cuticle that deters insects and microbes. Some plants go a step further with painful structures to warn would-be predators. Thorns, spines, and prickles discourage bigger herbivores. If these external defenses are breached, the plant immune system springs into action. Plants don't have a separate immune system like animals. Instead, every cell has the ability to detect and defend against invaders. Specialized receptors can recognize molecules that signal the presence of dangerous microbes or insects. In response, the immune system initiates a battery of defensive maneuvers. To prevent more pathogens from making their way inside, the waxy cuticle thickens and cell walls get stronger. Guard cells seal up pores in the leaves. And if microbes are devouring one section of the plant, those cells can self-destruct to quarantine the infection. Compounds toxic to microbes and insects are also produced, often tailor-made for a specific threat. Many of the plant molecules that humans have adopted as drugs, medicines, and seasonings evolved as part of plants' immune systems because they're antimicrobial or insecticidal. An area of a plant under attack can alert other regions using hormones, airborne compounds, or even electrical signals. When other parts of the plant detect these signals, they ramp up production of defensive compounds. And for some species, like tomatoes, this early warning system also alerts their neighbors. Some plants can even recruit allies to adopt a strong offense against their would-be attackers. Cotton plants under siege by caterpillars release a specific cocktail of 10 to 12 chemicals into the air. This mixture attracts parasitic wasps that lay eggs inside the caterpillars. Plants may not be able to flee the scene of an attack or fight off predators with teeth and claws, but with sturdy armor, a well-stocked chemical arsenal, a neighborhood watch, and cross-species alliances, a plant isn't always an easy meal.
There's so many things you can do with fungi, and this is what keeps us up at night. Fungi for food, medicine, textiles, fiber, packaging materials, even biofuel. Fungi just have this potential to unlock biological material that's a waste product in our civilization and convert it into something else. Whatever color. I have a lot of species of fungi here in the lab, and there's 1.5 million species plus of fungi on the planet.
Effectiveness, do we need to concentrate it somehow? Can we store it? Kind of my plan. Our theory is that when you have an infection and you're either not sure what the bacteria is, or you know that that bacteria is drug resistant, so we don't have a really perfect treatment, that you could take that bacteria, inoculate the mushroom, so put it on the mushroom would then sweat out these metabolites, and those metabolites would basically be able to kill that bacteria. So it's like a personalized antibiotic production system for your infection. Knowing the problems we have with antimicrobial resistance, something like this that's coming out of a different perspective and saying, look, let's take advantage of what the natural world can already do, to me, that's just fascinating. I think it's unlimited what we could do with mushrooms once we know more about the different varieties that are out there and more about their biology in general.
At this point, I hope you understand and somehow appreciate natural products already. Now, let's check to see whether you can differentiate primary metabolites from secondary metabolites. Which of the following are primary metabolites and which are secondary metabolites? Okay, let me reveal the answers. Guanine is one of the nitrogenous bases in DNA and RNA, so it is a primary metabolite. Tyrosine is one of the essential amino acids to make proteins in our body, so that is a primary metabolite as well. Glucose is the main source of energy of so many living organisms, and cholesterol is found in the structure of cells and tissues. So these four are essential in growth and development and can be found in a lot of organisms, so these are primary metabolites. Carotenoids are pigments that are found in only certain plants. These protect the plants from harmful UV rays and give them a bright orange color to attract pollinators. Morphine. We don't exactly know why poppy plants produce morphine, but we all know that this is a powerful painkiller for humans and can even get you high when in dangerous doses. And lastly, caffeine. It serves as a natural pesticide and can kill insects that ingest caffeine-bearing plants. Okay, next, NatProd focus secondary metabolites. So let's just discuss this briefly. The field of natural products in chemistry focuses mainly on secondary metabolites, although meron ding primary metabolites na nasasama. Now, bakit secondary metabolites ang focus? Well, primary metabolites are common within living organisms and they already are well studied to support basic life functions of organisms. On the other hand, secondary metabolites are found only in certain species. They also prove to be useful in medicine and other applications. Throughout the ages, humans have relied on nature for medicinal needs. From traditional medicine to modern day science, we kind of take advantage of the biological activities of these secondary metabolites. Okay, now let's go to the sources of secondary metabolites and some examples of their uses. There are plant and non-plant sources, so let's go through these. Plants. We already mentioned caffeine, carotenoid, morphine, but there are so much more in this list. Most of the natural products we gather are from plants because they are relatively easy to collect and grow, and there are just simply so many plants out there in nature that people are interested in studying. You will see more natural products from plants as we go along. Okay, next, fungi. Cephalosporin are antimicrobial substances used to manage a wide range of infections from gram-positive and gram-negative bacteria. These are useful against skin infection-resistant bacteria, meningitis, and other infections. This is derived from the fungus Acremonium, which was previously known as Cephalosporium. Next, epirubicin. Epirubicin is from the soil fungus Streptomyces, a chemotherapy drug primarily used against breast and ovarian cancer, gastric cancer, lung cancer, and lymphomas. Penicillin. Penicillins are a well-known group of antibiotics originally obtained from Penicillium molds. Alexander Fleming accidentally discovered penicillin when he left a culture of Staphylococcus bacteria during a 2-week vacation. When he came back, na-contaminate yung kanyang bacterial culture with fungi or molds, but in a moment of sheer serendipity, na-discover niya na kaya palang mapatay ng secondary metabolites ng penicillium molds yung Staphylococcus. Okay, lastly, streptomycin. This works well against bacteria that causes tuberculosis and works against other pathogens as well. Next, marine organisms and in particular sponges. So, some of the compounds from marine sponge are spongothymidine, spongouridine, and discodermolite. These have antiviral, anti-cancer, and anti-tumor properties. Okay, animals. Epibatidine is obtained from skin of the Ecuadorian frog, which is poisonous. It is 10 times more effective than morphine. Toxins and venoms obtained from animals played an important role in the curing of several diseases. For example, teprotide extracted from the Brazilian viper. It served as a lead compound and led to the development of the drugs captopril and cilazapril, which were effective against hypertension. The Philippines is rich with endemic plants and microorganisms that could be potential sources of useful natural products. That is why DOST is currently holding a program called Tuklas Lunas. Lagundi and Sambong medicinal products were developed under DOST, and now they continue to fund projects like these to eventually help discover new medicinal drugs. Isipin nyo, we have all these indigenous plants in the country and even weeds that grow randomly on the streets. These are all potential but untapped sources of natural products. Sadly, there are not enough researchers studying them. So, ayun, a little something to think about. Now, let's move on to the types of natural products. There are 3 major groups of natural products. One is terpenes, second phenolics, third nitrogen-containing compounds. For each type, we are going to discuss the general structure, the synthesis or synthetic pathway, the further classification or subcategories under that major group, and some examples and their general functions. So let's start. Terpenes. What is their basic structure? They are made up of isoprene units. Isoprene is a common name for 2-methyl-1,3-butadiene. So it's a conjugated diene. It has 5 carbons, as you can see. Isoprene units combine with each other to form larger structures of terpenes, and usually divisible by 5 ang number of carbons in terpenes. For example, limonene, found in citrus fruits, is a type of terpene. It is made up of 2 joint isoprene units. So you can look at the 2 isoprene units. One is colored red and one is colored blue. Okay, now, how are terpenes synthesized? There are 2 synthetic pathways for terpenes. We have mevalonic acid and methyl erythritol phosphate pathways. Both MVA and MEP pathways are found in plants, meaning plants can synthesize mevalonic acid as an intermediate for terpenes, but they can also synthesize it through producing methyl erythritol phosphate. The MEP pathway is found in chloroplasts, so eubacteria and green algae also use MEP pathway. On the other hand, animals, fungi, and archaebacteria use MVA pathway. Okay, next, how are terpenes classified? Ano ang subcategories ng terpenes? First, we have monoterpenes. Most of the time, it has 10 carbons, meaning it is made of 2 isoprene units. However, sometimes, along the synthetic pathway, pwede siyang mabawasan o madagdagan ng 1 or 2 carbons. So, for example, alpha-phelandrine has only 8 carbons. Next, we have sesquiterpenes, meaning they have 15 carbons, more or less, or 3 isoprene units. You can count the carbons in our example, alpha-selenine. It has exactly 15 carbons, making it a sesquiterpene. Next, diterpene with around 20 carbons. Next is triterpene with around 30 carbons. You might be familiar with the structure of the triterpene shown. Okay, you might have encountered this in Chemistry 3 as a kind of lipid, which is a sterol. Sterols have this signature structure wherein merong magkakadikit na 3 6-carbon rings and 1 5-carbon ring. So, don't forget, sterols are under triterpenes. Tetraterpenes have 40 carbons, just like this beta-carotene. Very visible yung mga isoprene units as seen in the conjugated pi bonds. Another term you might encounter are terpenoids. Some samples shown in the previous slides contain oxygen, right? Well, sometimes terpene derivatives contain oxygen and these are called terpenoids. But most of the time, terpenoids and terpenes are used interchangeably. Another type of terpene is saponins. These are terpene structures with sugar molecules. So, pag ang sugar molecules nakakabit sa metabolite, it is called glycoside. So, you might want to review the structures of sugars so you can easily identify them in large molecules. Okay, now let's go to some examples. So, try answering these questions. What type of terpene is shown on the right? This is a diterpene. It has 20 carbons. Next, what type of terpene is shown on the right? This is a diterpenoid. it has 20 carbons with an oxygen. Next, what type of terpene is shown? This is a triterpene. If you count the number of carbons, you will get 30. So this compound is an important precursor for the sterols, as you can see in this example here. So this is just the reaction mechanism of how squalene can be turned into a sterol. Okay, lastly, ano yung functions ng terpenes? So some terpenes have roles in growth and development. So some are considered primary metabolites such as gibberellins. And another function, terpenes defend many plants against herbivores. So terpenes can be toxic and can be feeding deterrents to many herbivorous insects and mammals. Okay, so next, now let's go to phenolics. Now, what is their basic structure? From the name itself, they suggest the presence of phenols, okay, which you will see later. But for a more generic structure, basta may benzene ring dun sa metabolite, it is considered a phenolic. So these are just some examples. So it can be as small as gallic acid na may aromatic ring with a carboxylic acid, or it can be as large as this structure on the right. Okay, now how are phenolics synthesized? So they can be synthesized via shikimic acid pathway or malonic acid pathway. The shikimic acid pathway produces most phenolics in plants, while malonic pathway is an important source of phenolics in fungi and bacteria. It can still be present in plants, but not so much common in higher plants. Okay, next, how are phenolics classified? Well, they can be categorized as benzoic acid derivatives. So, a benzoic acid derivative has an aromatic ring with a 1-carbon side chain. So, pwedeng magkaroon ng other functional groups like OH groups or carboxylic acid, but it follows the base structure or the skeleton of a benzoic acid derivative. Okay, next, simple phenylpropanoid. So the base structure of simple phenylpropanoid is an aromatic ring with a 3-carbon side chain. Okay, chalcone, the base structure of a chalcone, it has 2 aromatic rings and it is connected via a 3-carbon bridge. Okay, next, coumarin. The base structure of coumarin is 1 aromatic ring and another ring that contains an ester. Okay, flavonoid. It has 2 aromatic rings and there is still a 3-carbon bridge, but you can see that there is a cyclic ether in the middle. There are a lot of types of flavonoids, so let's just go through these. Meron tayong flavanone, so it has a ketone group in the ring labeled letter C. Flavanonol, meron tayong ketone and alcohol group sa ring C. Flavone, we have a ketone and an alkene. Flavonol, meron tayong ketone and an enol. Isoflavone, you can notice that the aromatic ring is displaced compared to the others. Flavantriol, meron tayong alcohol group sa carbon number 3. And lastly, anthocyanidins. We have a charge on the oxygen. So this is an ionic compound. Anthocyanidins are positively charged. These are some examples. So we have here an example of a flavonol, but then we also have here the same structure but with a molecule of sugar. So flavonoids can also have glycosides in their structure. Okay, next, tannins. Tannins can be hydrolyzable tannins or condensed tannins. Hydrolyzable tannins have a sugar molecule in the middle, and this sugar molecule is surrounded by gallic acid groups. Condensed tannins, on the other hand, are polymers of flavonoids. So you can see the structure of a flavonoid there that are connected with one another to form a larger molecule. So that is your condensed tannin. Okay, now let's try some examples. What type of phenolic compound is shown on the right? This is a flavonoid. So specifically, it is an isoflavone. Next, what type of phenolic compound is shown on the right? Okay, this is a flavonoid, particularly an anthocyanin. Look at the formal charge of oxygen there. Next, what type of phenolic compound is shown? Okay, this is a tannin. Specifically, it is a hydrolyzable tannin with a sugar molecule in the center. Okay, what are phenolics for? Well, they have a lot of uses. They can limit the growth of other plants. They can be colored, attracting other animals or such as pollinators. They can protect against UV light damage, and they can deter feeding by herbivores. Okay, lastly, let's go to nitrogen-containing compounds. Nitrogen-containing compounds have very diverse structures, but there is only one thing in common with them. They have nitrogen. Nitrogen-containing compounds can be synthesized via shikimic acid pathway and tricarboxylic acid cycle. So amino acids are the intermediates for producing nitrogen-containing compounds. So for aromatic amino acids, they come from shikimic acid, while aliphatic amino acids come from tricarboxylic acids. Now, how are the nitrogen-containing compounds classified? Well, first is an alkaloid. So alkaloids can have a lot of subcategories, but one thing is common in them. They have a heterocyclic ring with both carbon and nitrogen atoms. Examples are caffeine, nicotine, and morphine. Next, cyanogenic glycosides. So cyanogenic glycosides have the nitrile groups and are bonded to sugars. So examples are linemarine and lot-australin. Trivia: cyanogenic glycosides are toxic, and these are found in a favorite root crop of Filipinos called cassava. Cassava, if not properly cooked, Hindi nabe-breakdown yung cyanogenic glycosides, and once na nakain yun, they are toxic. Okay, next, glucosinolates. Glucosinolates contain nitrogens as well as sulfur atoms. Next, non-protein amino acids. The characteristic structure of amino acids have NH2 and carboxylic acid bonded to one carbon. So you can see there is a chiral carbon bonded with NH2 and COOH in our example. Okay, now let's proceed with the questions. What type of nitrogen-containing compound is shown on the right? Okay, it has a nitrile group, so this is a cyanogenic glycoside. Next, what type of nitrogen-containing compound is shown on the right? This is an alkaloid. Look at where the nitrogen atom is. It is part of a ring. So that is the characteristic structure of alkaloids. So overall, alkaloids are poisonous or they have physiological effects. So usually ang purpose nila ay pang-deter ng predators. So to recap, in Natural Products Part 1, we discussed primary versus secondary metabolites. We explained why secondary metabolites are focused in NatProd studies. We also talked about sources and examples of secondary metabolites, and we discussed the types of natural products. That is all for this video. Thank you for listening.